Hello class and welcome back for our third lecture video here on Unit 6 in regards to cell division. In this lecture video we're going to go through the first half of what we refer to as the cell cycle. So let's get started. So the definition of the cell cycle is going to refer to the period of growth and division of, remember from our last lecture video, of a somatic cell. So that's any cell in your body that's not a sperm or egg cell. So the lifespan of a somatic cell is put overall into, or classified into, two general periods: interphase, and then the actual division phase, which is made up of both mitosis and the last part of division referred to as cytokinesis. Alright, in this video we're going to focus more on interphase and all its smaller components. So interphase is also referred to as the growth period of a cell. So if you look at this little animation up top on the right, it's the point where you have a brand new baby cell here, and it starts to grow and get bigger until it has to copy its DNA and eventually divide into two brand new cells. That's the key. All right, it's going to take up roughly 2/3 or more of a cell's lifespan. So what's 2/3? About 66.666, etc. %, right? I would say that you want to see cells spending at least 60% or more. So 2/3 is a little bit more, but if you were just round down to about 60, I would want to see a interphase value of 60% or more of a cell's life, a specific type of cell's life, is spent in interphase. Then the last third or even less of the cell's life should be in actual division or mitosis, where you're going to create two daughter cells like you see up here. All right, so we're looking for, again, a third or less of the cell's lifespan should be in some stage of mitosis, which, if you remember from 7th grade, are prophase, metaphase, anaphase, and telophase, and then cell division ends with officially cytokinesis. So let's look at the subphases that make up interphase. So interphase is broken down into three smaller subphases. We have G1, S, and G2. All right, the G stands for gap, or I usually refer to it as growth. So when a cell is born, you get a brand new little baby cell. It enters officially G1. And during G1, pretty much that cell instantly starts to grow rapidly. Now, that pace is going to differ from cell type to cell type, but they're going to start to grow. And as they grow, they're going to go about doing whatever their normal function is. However, and we discussed this in our diffusion osmosis unit, cell size is limited. A cell is going to get too big at one point, and once it gets too big, its rate of both diffusion and osmosis is going to start to significantly suffer or decrease, and that could lead to cell death, either by starving to death or it can't get rid of its toxins fast enough. So at some point, that G1 cell is going to grow too big and pass something that's referred to as the restriction point, sometimes also called the point of no return. So this restriction point, which is going to be at some point in gap 1 or G1, is where the cell has now officially gotten too big and its rate of diffusion is going to suffer drastically. So the cell has two options at that point: divide if it can, or die. Just get out of the way and make room for another healthy cell. If the cell can divide, it officially begins what's called the S phase, which stands for the synthesis of all its DNA and chromosomes. So during the S phase of interphase is where all the DNA inside is actually going to get duplicated. And then the third and final subphase of interphase is G2. At this point, all organelles within the cell are going to be copied. You name it. Centrioles, which we're going to discuss for mitosis, mitochondria, rough ER, smooth ER, Golgi. If it's a plant cell, chloroplast. Every single organelle will have numerous copies of itself made. The cell will grow even more and produce numerous proteins that are going to be needed to drive mitosis, and proteins that will be needed for both brand new baby daughter cells. So again, if you look up here in gap 1, this cell has already gotten too big. But look at the size of it now in G2. It looks like it's grown at least around 30 to 40% more than it was originally. Because the idea is you're going to cut that volume in half once this big cell does separate into two smaller brand new baby daughter cells. You can see, hopefully, in the image that here are the normal chromosomes, which, by the way, you would not see until mitosis anyway, but they're there. They've already gone through division during the S phase, so you can kind of make out that they now have little X's. They look like X's and are connected. They get connected together by what's called a centromere. And before mitosis actually does begin, the last thing that needs to happen in G2 is all of the DNA and all the chromosomes that were copied up here, they need to be double checked for errors. All right, and you're going to see why there's going to be many chances for errors once we go and learn about in subsequent lecture videos on especially the process of DNA replication. There's a lot going on and a lot of chances that errors could in fact occur. Most of those errors are found and corrected before division takes place. You don't want those errors to pass on to new cells that would then have those errors permanently and continue to pass those on. If those errors are found, they'll be fixed. If they can't be fixed and they are found, usually the cell will be destroyed just to avoid any mutations being passed on. But obviously that does happen occasionally, and DNA mutations are what are the result of us aging right now currently, and that can lead to diseases as a result of aging, such as cancer. So again, interphase, over 2/3 of a cell's life, or hopefully at least around that much, is spent in these three subphases making up interphase. If we look at just a graph of it, all right, no matter, every picture might look a little different. I would always try to find mitosis first, which you're always going to see represented by many smaller little pie pieces or little tiny slices of pizza, you could think. So if M phase is up top, on the right, down below, on the left, it doesn't matter where it's at. I just need you to realize no matter where it is, if you go clockwise right after it, this first larger pie piece is going to represent G1. Again, which is the growth and overall preparation for DNA replication if the cell can in fact replicate itself. After the cell gets too big, so this dotted line I put here represents the restriction point. If it does pass that, the restriction point always has to be somewhere in G1, because a cell is not going to start copying its DNA here in the S phase unless it's already past the replication point. Synthesis of DNA would then occur. Cell enters G2 where you get more growth. Preparation for mitosis then would be producing excessive amounts of proteins, copying all the organelles, and then obviously proofreading, double checking the DNA for any errors or mutations before you would enter official mitosis that we'll get to later in another lecture video. Now, if you notice, I have something called G0 over here too. It's kind of like a satellite, kind of like the moon orbiting the Earth, right? But it's orbiting G1. G0 refers to cells that never reach the restriction point and hence never divide. So what that means is if you lose them, they're gone forever. So there are some cells in your body, once you reach the age of around 11 to 13, that they stop growing, which means they're never going to pass this restriction point. And if they never pass the restriction point, they never go through the rest of the cell cycle to get to division. So they're kind of in limbo, these G0 cells. They're never going to grow to pass the restriction point to go through division. So if they get injured or killed, they're gone forever. So some examples of these are as follows. Heart, brain, and nerve. Obviously, you've developed into the person you are, so all of these cell types would have had to have grown at some point. But once you reach your overall developmental peak, right, particularly for your heart and brain around the ages of 11 to 13, they don't really grow much more at that point. Nerve cells still may grow until you stop growing, and then they're going to shut down, at least the main ones, they're going to shut down from growing or expanding also. So at that point, if you lose them, they're gone forever. So that's what I mean by G0 cells. Now, why do they do this? I'm not going to hold you accountable for this, but your brain, think about your brain and your skull. There's only a little itty bitty space between the edge of your brain and your skull. There's no room for it to expand. If it were to expand, it would hit the skull, which would put pressure back down onto the brain and cause potential brain damage. That's what a concussion is. Your brain swells, the swelling then pushes back down on the brain, and you do get some mild brain damage. Even if you're fine, there technically is brain damage. Your heart's a muscle, all right? Your heart needs to be able to be fit, but pump really easily too. If your heart were to get too big, it'd be like a huge bodybuilder who can't really stretch too much. He tries to flex or show you their muscles, but they're not going to be touching their toes necessarily or anything like that. If the heart gets too big, it won't be able to flex and pump properly, so it could go into arrhythmia and your heart would stop. And finally, your nerve cells, well, they conduct electricity, particularly from your brain, down your brain stem, your spinal cord, to all your limbs. Think of them as electrical cords that you would plug into an outlet at your house. You're only supposed to put in a couple, even with an extension, like an extension plug cord into an outlet in your house. Should you plug in like 10 or 15 separate items into that outlet? No. It's going to cause a spark or break this or trip the circuit that's in your house. Well, if you have too many nerves, that could drain the electrical impulse coming from your brain, which could delay the messages that your brain are sending to their destination. So think about your heart. Your brain is constantly telling your heart to pump. If there's too much of a drain on the electrical impulse because you grow too many nerves, your heart may not get the message to pump fast enough, and instead of going ba-boom, 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 it starts going ba-boom, 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 and it forgets the signal and it stops. So these cells need to stop growing for the overall benefit of us or the organism. And let's finish up with some vocabulary here that we're going to see come mitosis. So on this page, I have what's called a chromatid and sister chromatid pairs. A chromatid is just an identical copy of a chromosome. So if you look down here, here's a chromosome. It goes through the S phase, and now you have two halves of that X. Each half is an identical version of that original chromosome called a chromatid. And as long as those two chromatids are connected in the middle here by what we call a centromere, that center part of the chromosome, that would then be a sister chromatid pair. So every X that you see represents a sister chromatid pair, or you could also say 1, 2 chromatids. All right, I would also call this one single chromosome. As long as each half is connected by one single centromere, always count your centromeres for chromosome numbers. So to finish out for our last slide here, on the left, I have 1, 2 chromosomes. I'm going to count my centromeres. Or I could say I have 2 homologous chromosomes, because you see how they're both the same size and shape, and we discussed homologous chromosomes earlier. So that means there's a version here from mom and dad for that one chromosome type. And if they're both homologous, containing the same genetic information as each other, I could refer to it also as one homologous pair. Now, after DNA replication during the S phase, we won't see again chromosomes until we hit prophase of mitosis, but this is how they would look at the end of the S phase as well as into G2. If you count your centromeres here, you still have 2 chromosomes, 1, 2. Or you could say you have 2 homologous chromosomes because they still contain the same genetic information. Or 1 homologous pair because they both have the same info. But now we're going to use some of those new terms. Now I have 2 X's, right? So I could say I have 1, 2 sister chromatid pairs. And then since each sister chromatid pair is made up of 2 chromatids, 2 identical versions of the original chromosome, I'm going to have a total of per X 1, 2, 3, 4 chromatids. So that's really the only hard part of mitosis, is getting these new vocab terms down and assigning the correct numbers to them.